in Dubai, and thank you for attending my Zoom presentation on the webinar session. I want to tell you about a new therapeutic approach for ASD, neural injuries and developmental disorders, which is based on low-level laser therapy. And as I didn't put a picture of the device itself, I brought it with me. This is how big it is, the piece I use for babies and toddlers. And let us start now. ASD displays early in childhood development during the time of intense human synapse formation and maturation, and usually results in long-term difficulties in social, communicational, emotional, adaptive, and cognitive functions. The frequency of ASD occurrence continues to rise from 1 to 110 in 2006 to 1 to 54 in 2016, with at least one diagnosed coexisting neurodevelopmental disorder in most of the children. Early diagnosis and treatment are very important as they may minimize neural injury and functional difficulty. And as ASD is still diagnosed only by behavioral criteria, it has been difficult to connect the numerous neurophysiologic findings to the clinical characteristics of ASD and to draw the mechanism and etiology of autism, until recently so. This would allow an accurate treatment directed to the mechanism of injury with the best chance to make a change in the impaired developmental route. The search for ASD brain mechanism may be reviewed almost chronologically from the neural circuit to the molecules and organelles that are involved. In the late 90s of the 20th century, a laboratory in Italy first documented neural activity from brain cycles, later named mirror neurons. The innovation in this discovery was that it connected field of neural control that were considered separate, motor and vision. That is, the same specific neural cycles work both when a person does something and when he or she watches another person perform the same action, making an instant translation from visual to motor control. This act of neural translation is considered the basis of the human ability to imitate, to anticipate others' goals, and to empathize others' pain or misery. These mirror neurons' brain cycles showed altered activity in children with ASD, hinting that they are involved in the mechanism of ASD. Now, the scientific findings about mirror neurons and the possibility that their development may be related to the time window of temporary subcortical plate neural circuits, which connect the thalamic and future cortical cycles early in fetal development, were indeed amazing. But the mechanistic discussion in the level of neural circuits left many open questions. What may cause damaging alterations in these brain circuits? What cellular and molecular components are involved, and how can we target the therapeutic process to them? Now, loss of synaptic stability and plasticity or dysregulation of activity-dependent signaling networks that control synapse development, function, and plasticity may cause injuries in neuronal circuits and contribute significantly to brain disease, including ASD pathogenesis. Hence, alterations in synapse functions, synaptic molecules, receptors, and neurotransmitters have been targets to research about the mechanism of autism syndrome for the last 20 years. Studies showed that alterations in glutamate receptors and enhanced GABA receptor-mediated inhibitory synaptic transmission are involved in autism. There may be various causes involved in ASD: genetics, drug use, neurodegeneration, viral infections, and more. However, dysfunction of neuronal synaptic communication is almost always the underlying cellular mechanism. Epigenetic changes in synaptic genes encoding for synaptic adhesion molecules like neurexin, neuroligin, and N-cadherin, and for PSD proteins like SHANK1, 3, and others are involved in ASD, causing alterations in synaptic transmission. And recent studies have found that failure of the cellular machinery in pathways upstream of synapse lead to synaptic dysfunction and neuropsychiatric characteristics. In addition, small non-coding microRNAs that repress the translation of target messenger RNAs seem to be important. Pathophysiologic mechanisms for neurologic and psychiatric diseases and abnormal regulation of protein turnover, chromatin remodeling, and genomic imprinting may lead to synapse pathology. In some neuropsychiatric disorders, the basic neurobiological mechanisms underlying the symptoms are simple and easily solved, but most neuropsychiatric disorders, including autism, are etiologically heterogeneous and complex and are likely determined by a combination of various variants, defects, injuries in multiple genes. For example, genome-wide association studies identified polymorphic variants in genes encoding synaptic proteins as important determinants of the risk of developing ASD. Recently, multiple studies show that a mitochondrial disease or abnormality is involved in the etiology of ASD, affecting about 80% of the children with autism. Mitochondria are the cell power plants, being responsible for most of cell energy production. And sufficient energy is required for everyday vitality and for brain survival and function. And brain cells specifically need a lot of energy for function. Apart from energy production, mitochondria participate in the cellular metabolic process of iron and the balance of calcium. The mitochondria are associated with normal and abnormal cell proliferation and participate in programmed cell death. Each cell has hundreds of tens of thousands of mitochondria, depending on the role of the energy consumption of that cell. Now, mitochondria are inherited only from the mother through the ovum. They can develop mutations as they multiply and lack almost any repair mechanisms. Most of the proteins that make up mitochondria are encoded in the nucleus, and as the mitochondria are inherited from the mother, hence we don't have a backup from the father's genome when mutations or damage occurs. But since the ovum contains a lot of mitochondria to start with, some may be damaged without any clinical manifestations. Mitochondrial damage may be manifested over the generations until there are only few normal mitochondria, and then a problem is revealed in such cases clinically. A damage to mitochondria may be caused not only by maternal inheritance, like when cells divide to form the fetus, but also by coding error called de novo mutation, when a new mutation in fetal cells or in mitochondria happens due to environmental or epigenetic influence. And mitochondrial abnormalities include either decrease or increase in mitochondrial function, and depending on the cause and developmental time window, When this happens, they may lead to neurodevelopmental regression and the typical comorbidities that are associated with autism, meaning gastrointestinal problems, seizures, tiredness, and sensory dysregulation. The first findings leading to this conclusion go back to the 80s of the 20th century, and then reconfirmed 20 years later, and continue with studies that examine accessible biomarkers of mitochondrial dysfunction until today. Neurodevelopmental regression, as typically described for many children with autism, may be the hallmark of a mitochondrial disorder and abnormal mitochondrial physiology in autism. And as mitochondrial function is highly influenced by environmental factors, these findings connect mitochondrial dysfunction and ASD with environmental hazards. Since autism was first defined, numerous treatments have been employed with partial and sporadic mechanistic justification. Most of the treatment approaches target behavioral abnormalities of children with autism and aim to improve the social and communicational function of the patients. The website of the American Association of Communication Clinicians describes 30 common therapeutic programs for children with autism divided into several classes. However, parents cannot be given definite treatment recommendations because of the heterogeneous characteristics of children with autism and because many therapies have not yet been investigated in a controlled and satisfactory manner. In November 2020, the Australian governmental CRC top organization published a 502-page document written by 12 scientists. The paper is a meta-analysis based on 58 review articles, analyzing more than 1,000 research articles that examined the effectiveness of 111 different autism therapy programs. The authors sorted the programs into 9 categories: cognitive, behavioral, educational, developmental, animal-assisted, sensory-based, naturalistic, technology-based, and more. The authors note that in each category, only a very small number of studies were made in a controlled and satisfactory manner, meaning that the results should be treated with caution. Altogether, children with autism can be treated in a way that will lead to functional and communicational improvement using various therapeutic approaches. These treatment plans are tailored to the unique behavioral profile of each child and each family at each point in time throughout their life journey with autism. However, as these treatments focus on external behavioral symptoms and not on the internal mechanism, and they aim at functional improvement and not actual repair of the neurological damage. Hence, according to this approach, autism is not a curable injury, but a developmental disorder whose treatment helps patients develop functional skills, improve communication skills, and rely on their strengths despite the disorder that will always remain a part of their lives. Now there's a question: should we be satisfied with this important achievement of symptom-oriented therapeutic approach? Or perhaps a persistent search into the mechanistic questions may lead to a mechanism-oriented therapeutic approach. This means going back to the mitochondria. Few therapeutic approaches for mitochondrial disorders were examined in clinical studies with children diagnosed with ASD. These include cofactor supplementation and ketogenic diet, nutritional supplements aimed to support the mitochondrial redox and folate pathways, and contained L-carnitine, coenzyme Q10, and additional factors. They improved mitochondrial function and ASD symptoms. However, discontinuation of the supplement treatment caused rebound effect and worsening of the ASD behavior in children. Ketogenic diet has been studied for autism, resulting in a mild to moderate improvement in 58% of the children who tolerated the 3-month-long diet. And in 1 of 3 studies, worsening outcomes were observed. In addition, the ketogenic diet improved sociability and repetitive behaviors in 2 environmentally induced mice models of autism. With these results, the ketogenic diet needs more study for its use in children with ASD. And in addition, the important limitation of the ketogenic diet and other dietary treatments for children is the ability of the family to implement the diet adequately and the child's ability to tolerate the diet, as dietary therapies are difficult to implement with children. Now, the hormone oxytocin has an important role in social behavior and involved in eye contact, emotion detection, trust, peer identification, and attachment behavior in humans and in mammalian species. Specifically, low brain levels of oxytocin have been found in autistic people. This has led to an assumption that administration of synthetic oxytocin may have a therapeutic potential in the treatment of psychiatric syndromes with social characteristics. Within few years, a new protocol developed, and intranasal administration of oxytocin has become the preferred route owing to it being well tolerated and easy to use. However, it was recently found that administration of external oxytocin causes neuromodulation and downregulation, like intolerance of natural oxytocin release, and there's no improve, opposite. But a novel therapy employing low-level laser therapy may cause long-term rise in natural oxytocin release. The most common therapeutic approach in Western medicine for various pathologies, also those with a mental, emotional, communicational background, is pharmacology, but it is only partially effective. In recent years, the use of therapeutic approaches that are based on ancient knowledge from East Asian cultures, combined with clinical, empirical, scientific knowledge, has increased. In the therapeutic approach described here, we employ an innovative and well-known and scientifically based tool with autistic children. Lasers, the abbreviation of light amplification by stimulation of radiation emission, are devices that produce electromagnetic radiation which has uniformity in wavelengths, phase, and polarization. A low-power laser is a special type of laser that influences biological systems by non-thermal means. In low-level laser therapy, blue and red and near-infrared light is used on injuries or over points on the skin that may influence the functioning of the brain and other organs in order to improve the healing of soft tissues, reducing inflammation, and relieving physical and mental chronic and acute pain. Low-level laser therapy applies a non-invasive therapeutic laser to the stimulation of specific points on the skin. This technique is considered safe and painless and has become an important tool for the treatment of patients at risk, such as premature babies. For example, stimulation of specific points for pain reduction using 3LT creates a local photochemical effect that causes specific changes in the activity of neurons in the brain, which is perceived by the patient as reduction of pain severity. And these changes were measured and quantified using imaging. Low-level laser therapy has a photochemical effect, meaning that when the correct parameters of power, wavelength, and location are employed, the laser beam reduces tissue oxidative stress and increases ATP levels. Lasers improve cell metabolism and reduce inflammation. And in addition, laser therapy has been shown to alter axonal flow and opioid receptor binding and endorphin production. And in clinical studies, it was found that laser therapy causes an immediate decrease in acute and chronic pain and can increase in cellular function. In addition, laser therapy has shown promising results for myocardial function, mesenchymal stem cell regeneration, skin injuries, brain trauma, TBI, diabetic retinopathy, oncology, and more. Infrared waves can increase the body's formation of ATP, DNA, RNA, the release of nitric oxide and cytochrome C oxidase, and the regulation and reduction of reactive oxygen species. And the change in the activity of the membrane of the intracellular organelles, mainly in the mitochondria, in calcium flux and stress proteins, and more. Laser therapy produces a shift toward higher oxidation in the total redox potential of the cell, and this change in the redox state of the mitochondria regulates several proteins in the cell, including cyclic AMP, CREB protein, kappa beta protein, hypoxia factor 1, and others. As a result, both ATP levels and blood flow increase, and we see an improvement in the availability of oxygen in brain damaged areas. The laser device has several therapeutically proven wavelengths: 405 nanometer, 670 nanometer, 808 nanometer, 950 nanometer. These wavelengths are within the optical window that allow optimal penetration depth with very low absorption of water, hemoglobin, and melanin. Laser source combined with a wavelength in the blue range, the 405 nanometer, was found to give best energy results, and hence, despite its low penetration capacity into tissues, a combination of a laser source with the blue wavelengths brings the best results in terms of energy and best clinical results. The blue wavelength is absorbed by the first complex of the cellular respiratory chain, the NADH, showing 5 times higher absorption than the red wavelengths. And it was found that with the help of bichromatic radiation, red and blue, one sees a greater change in the oxidation potential, redox, in the mitochondria and cytoplasma by oxidation of NADH. This increases the proton motor force that drives the flow of protons into the mitochondrial matrix and thereby increases the ATP cycle. In addition, the transfer of electrons is accelerated, and both effects cause an increased synthesis of ATP. To summarize, a wide range of seemingly unrelated disorders such as schizophrenia, bipolar disease, dementia, Alzheimer, epilepsy, migraine, strokes, neuropathic pain, CP, TBI, Parkinson's disease, ataxia, diabetic retinopathy, transient ischemic attack, cardiomyopathy, coronary artery disease, chronic fatigue syndrome, fibromyalgia, SARS-CoV-2, and more have common underlying pathophysiological mechanisms namely production of reactive oxygen species and the accumulation of damaged mitochondrial DNA, resulting in mitochondrial dysfunction. Laser therapy has been long recognized as an efficient therapeutic tool for brain injuries, including autism, and recent discoveries of the role of mitochondria in autism etiology and in the laser therapeutic process gives us a great opportunity to improve mitochondrial function and brain neural development using laser therapy energy on specific ear and body skin points. Thank you very much for your attention.